Our final speaker of this session is Michael Snyder. Dr. Snyder is the Sanford Asherman Professor and Chair of Genetics and the Director of the Center of Genomics and Personalized Medicine at Stanford University. And he will talk about the integration of multiple omics.
All right. Well, my thanks as well for having me here. It's really terrific to learn all this wonderful research going on. I'm going to continue on this theme of trying to use individualized profiling, if you will, understanding the individual to better manage people's health. And we're very focused on big data of all types, as you'll see in a minute, to do just that. And these are my conflicts here. I have many conflicts. You can just trust everything I'm about to say. Two in particular, Qbio is a big data health company, and January AI is a CGM metabolic health monitoring company. So I'm sure you all know your health is a product of many things, your DNA, as you've heard earlier, but also the various exposures you might encounter and all kinds of lifestyle things as well, stress, food, exercise. These all impact your health. We know this intuitively, but we're entering a world where it's now possible to quantify this. We can certainly sequence people's genomes, and we can measure these various things to different degrees of accuracy. Some of it's not so bad, like activity. Some of it's still a bit clunky, like food logging. But nonetheless, we can quantify this. Perhaps more importantly, we can quantify its effects, the effects of these various things, by taking very detailed molecular and physiological measurements on the person itself. And so a number of years ago, we set up something we call personal omics profiling, where the idea is to do very, very deep measurements on people. We would sequence their genome once, but then from their blood and their urine, especially peripheral blood monocyte cells, immune cells, we would make all these omics measurements, if you will. This is DNA methylation. This is following people's RNA, proteins, critical immune molecules, metabolomics, lipidomics, and so on. And the microbiome, as you've heard earlier. So we'll take very, very deep profiling. This come out of nasal and stool, now more recently other areas. We also do deep clinical testing, questionnaires, and some advanced tests as well. And I'll tell you some of our work using Wearable. So we're collecting very, very deep measurements on people. And the other aspect is we're following them longitudinally. So we'll profile people, we'll sample them roughly every 3 months while they're healthy. And then if an adverse event comes along, like a viral infection or perhaps other health challenges, we'll take more samples, typically 5 to 7 additional samplings. And so we've been running this study, to be honest, for a little over 8.5 years, some a little more, some a little less. I'm the guinea pig who started. I've been profiling me for 11.5 years now. And so you might say, well, why are we doing all this? Well, we're trying to understand what it means to be healthy. What does a healthy profile look like? How does it change over time? How does it differ between different people? And what happens at the earliest times of disease, say with a viral infection, very relevant to today's world. And perhaps lastly, we wanted to see whether these advanced technologies like genome sequencing and some of these deep profilings, which we do with mass spectrometry and other methods you've heard about from others speaking so far, can these actually be used to better manage people's health? And so just getting to the last point, just from the first 3.5 years of profiling people, we had 49 major health discoveries. Now, it is an older cohort, 53.4 average entering the study, but it's a pretty wide range. And anyway, these health discoveries cover a wide range of areas: hem-oncology, cardiovascular disease, metabolic health, and so on and so forth. Quite a few different areas. And some of these discoveries were a big deal. So, for example, we caught someone with early lymphoma, 2 people with pre-cancers, MGUS, smoldering myeloma that can convert to aggressive cancers, 2 people with serious heart issues. This one here, actually one picked up with wearables, and that's a key feature. No one technology found all these things. It was often, sometimes genome sequencing, sometimes it was imaging, sometimes it was molecular measurements, either RNA or other metabolomics or what have you. Sometimes it was wearables that would uncover this. And often it was multiple variables that said something was up. And what was also important is that in nearly every case, in fact, in all cases, these are found pre-symptomatically. So we actually found these by making measurements on people, not because they had symptoms already, but we uncovered it through this deep profiling. And just to show you a few examples of genome sequencing, 13 people learned something pretty important from their sequence, 12 from Mendelian what are called Mendelian situations like bracket mutations. And some of these were a big deal. One individual, a young person, had a mutation in a cardiomyopathy gene, and turns out he has a heart defect, which was uncovered from this. Turns out it was running in his family as well. Another individual had a mutation that put them at risk for certain kinds of cancers, and they did a whole body MRI follow-up, and sure enough, they had an early thyroid cancer, which was removed, and they were able to keep their thyroid and don't need replacement therapy. And then 9 individuals coming in the study were diabetic. They were all thought to be type 2, but one of them turns out to be a MODY and was on suboptimal medication for years, actually. So genome sequencing was powerful. Imaging was powerful. That's how we picked up the lymphoma. That plus a couple of blood markers were elevated. This is the pre-cancer. This was picked up because one individual, also fairly young, they given 2 samples. Their 2 samples were just so different from everyone else's. There's 1,000 samples here, but this person's IgM was very, very elevated in this individual relative to everyone else. And then follow-up said, yep, they have too much IgM overproducing cells. Some people had plaques in their arteries on one of them. We have a lot of interest in type 2 diabetes because I'm actually type 2 diabetic, which my genome predicted, interestingly enough. And just to illustrate this point, coming into the study, this is what people thought they had. 9 individuals were thought to be diabetic. 2 were diabetic who didn't know it. And of course, a ton of people are pre-diabetic who didn't know it. You may know 9 out of 10 people are pre-diabetic, don't know it. And this is a big deal because 70% of those will go on to become type 2 diabetic in their lifetime. So we think it's important to find this. And actually CGM, continuous glucose monitoring, we think is powerful. We're very interested in how people become type 2 diabetic. 9 became clinically diagnosed as we ran the study, and 9 individuals became clinically diagnosed as diabetic, and others had diabetic range values. And it turns out people became diabetic in different ways. 2 people got there by weight gain. The other 7 didn't gain weight. And 5 of the 7 just gradually got there through elevated fasting glucose, or something called an oral glucose tolerance test. 2 people, it's as though something triggered that. I'm one of the 2. This is me, 8 years of my data. I first became diabetic after a very nasty respiratory syncytial virus infection. I got it under control, although it did come back later. It's kind of a long story, but I'm a very unusual diabetic, and it was actually data that helped me figure it out. I don't release insulin from my pancreas right. I'm a different form diabetic, and there's a specific drug, I'm a metformin non-responder, which is a frontline drug. I actually respond well to something called rapinolide, which makes sense for people who don't release insulin from their pancreas. So we actually think diabetes is really a heterogeneous set of disease, especially type 2 is probably really 50 different diseases. And by actually better understanding this, we could actually better manage people's health. So we call this precision diabetes. We can give a whole seminar on that. The other thing we've learned as we've gone along is that everybody's aging differently because we're following people's molecular changes over time. What we've discovered is that from 2 years of data, you can see how people are aging if you have 5 or more measurements, and everybody's aging differently. I'm a pretty typical ager. My coagulation, metabolic, and other pathways go up. Not so much my immune ones, which is pretty common. So at least that one's under control. But this other person, their top pathway changing is a cardiac hypertrophic signaling pathway. They're a cardioager. We can actually start grouping people into ageotypes, if you will, how they're aging. And so each column's a different person. Each row is one of these 4 ageotypes. We think there are more than 4, but this is how many we can follow readily. And so these people over here, they're aging in all 4 categories. This person's aging in 3 of the 4. This person's mostly a kidney ager, kidney and metabolic, and so on. So we can see how you're aging over time. So we think of it like a car, that as your car gets older over time, but some parts wear out first, and you can flag this by doing these deep profiles on people. One way in which we're thinking of scaling this, or are scaling this, is we've founded a company. I'm in Silicon Valley. What can I say? And the same thing as we're actually doing a medical version, as what I showed you before, is research, but you can actually do a more medical version. This one also has whole body MRI, and the same principle held. Just from the first 100 plus people, we learned a lot of pretty important things. In most cases, it was multiple measurements that said what was going on. So, for example, we caught some early prostate, ovarian, cardiovascular disease, so on. And in some cases, it's the longitudinal profiling that's very, very important for finding this, seeing this change, seeing things shift up. And I think that's underappreciated in today's world, that these longitudinal measurements are really, really critical for trying to follow people's health and what their health trajectory looks like. We're very, very keen on wearables. This, we think, is a fantastic way for looking at physiology. We got involved in this, but most people are using these as fitness trackers. You may know that wearables are getting to be very common. 20% of the US, 50 million people wear a smartwatch. Why they're powerful is they make hundreds of thousands of measurements on you every day, 365 days a year. Some of the devices will even make 2.5 million measurements on you each day. And they're capable of tracking many things. Nearly all of them do heart rate variability. Some do respiration. Some do skin temperature, blood oxygen, blood pressure. All these can be measured with a smartwatch. And then there are other devices as well. You're getting a whole lecture, I'm told, about continuous glucose monitoring. We're very, very keen on this. We think this is super powerful for understanding nutrition and people's response to this. And as I say, so we've formed a company around this. What's powerful about these physiological measurements is you can see when people are getting ill. In fact, we discovered you can detect infectious disease from a simple smartwatch and initially from a smartwatch and a pulse ox. And in fact, I identified my Lyme disease when I first got it pre-symptomatically from a smartwatch and a pulse ox. And bottom line was I could see this is resting heart rate, this is skin temperature. I'm not shown here as the pulse ox, but when I first got Lyme disease, I had basically my resting heart rate shot up as actually on an airplane. It will shoot up normally on an airplane, but it shot up more than normally, and my blood oxygen dropped more than normally, and they never returned to normal when the plane landed. And so we looked over the data I had at the time. I had 4 years of data. I was actually ill 4 times, the Lyme disease, and then 2 viral infections here and here. And the 4th time I was asymptomatic, but I was ill, presumably because I had C-reactive protein levels just as high as these other times. And every single time I was ill, you could pick this up by outlying resting heart rate measurements and skin temperature. And so we went on to write an algorithm to be able to detect when people get ill from this jump up in resting heart rate. And it actually works. It worked on me. This is all retrospective data, but every single time I got ill, this is a delta plot, a change from baseline. You can see this prior to symptom onset. It worked on me. It worked on 3 other people, one of whom got sick twice. So the point is you can actually see when people are getting ill from a smartwatch because their resting heart rate jumps up. So as you might imagine, when the pandemic hit, we were already working to improve these algorithms and actually build a system that could go to scale to literally do this on millions of people. And so, as you know, we're in the middle of this well, I don't know if we're in the middle. We'll see where we're at in this pandemic. This number is out of date. We're already over 100,000 a day again from this. And so one could argue, rapid detection is a very, very key component of all this. And the way we do it these days is with thermometers, which I would argue is a 300-year-old technology and not nearly as good as a smartwatch if you get right down to it. And it also, PCR testing is, of course, good, but it takes a day to get back your results. And it's expensive, and people do it stochastically. So the power of using wearables is that we think we can actually measure physiology in real time and detect changes pre-symptomatically. And so to test this idea, when this pandemic hit in March, we basically launched a new IRB study. We had one running, but we had to modify it. Launched it, quickly enrolled 5,000 people, 32 of whom had gotten COVID. They had heart rate data from a Fitbit, and they also had a diagnosis date and a symptom date. And this is our very first case we had. This is looking at resting heart rates, a standard deviation plot. This is the diagnosis day here. This is the symptom day. And what you'll notice is that this person's resting heart rate jumped up 9.5 days prior to symptom onset. And you can't miss the signal. It's very, very clear. And we've written algorithms, I'll show you in a minute, that can pick this up. This is a real-time monitoring system. Again, this one's retrospectively. And by the way, we're always looking for more participants. Feel free to join our study. If you have a smartwatch, all folks are welcome. From the first 32 people, we could detect, it turns out, 80% at or prior to onset of symptoms. And it turns out the median is 4 days prior to symptoms. So we can see a jump up a resting heart rate. We've written two different algorithms, works both ways, where we'll see this jump up in heart rate 4 days prior to symptom onset, 7 days prior to diagnosis. And it's not specific for COVID. Other illnesses will trigger this as well, trigger this elevation of heart rate. There, it's 2 days prior to symptoms. And that makes sense because COVID has a longer pre-symptomatic period. And this is not some giant jump up in heart rate. The median is 7 beats per minute, but for some people it's as little as 2 beats per minute, which again, you can pick up because you're measuring people 24/7. So we've gone on, we first wrote a real-time detection system for following this and showed it did work retrospectively. And now starting last December, beginning of December, we launched a real-time study. And again, love to have you sign up that will send you alerts when we detect this resting heart rate. We set it in a way that the alarms go off. We're trying to maximize sensitivity, but then also not have too many alarms. So the alarms we set to go off roughly every 6 weeks to 2 months, depending on the person. And they'll send a red alarm off. We have 3 different algorithms now. This one we set up in a way because it's actually not computationally intensive, and we think we can scale it to the planet. We think we can measure literally hundreds of millions of people with the system we built. It's a cloud-based system. We have an app. So basically you download it, it's all free. And then from the first 68 cases, now we're up to 84, and it's 80% of the time we can detect when people are getting ill at or before symptom onset because of this jump up in heart rate. And actually it brings in steps and other information to account. It not only works on pre-symptomatic cases, it does work for asymptomatic cases. So here are 2 examples here. This individual, there's when they're diagnosed, again, they didn't have symptoms, but their heart rate jumped up well in advance of this. I think this one's 14 days earlier. Here's another case where a person had it. I forgot to say, but our system works well for Apple Watch and Fitbit. I'm sure it'll work for Garmin too. We just don't have as many signups for that. And we can pick up these asymptomatic cases here, and it seems to work pretty well. I don't think these devices are going to be just for picking up infectious diseases. We can show using machine learning and AI that we can detect some clinical measurements, if you will, from a simple smartwatch. And this is with a fairly old device. I think we can do even better now. But you can get a signal about people's hemoglobin levels, red blood cell counts, hematocrit from a model based on wearable data. We can also get same information about hemoglobin A1C and glucose, fasting glucose, and some other measurements as well. Again, these aren't clinical diagnosis grade measurements, but what they're good enough for doing is giving you a clue that something's off. And that can be powerful for detecting anemia. You know, when people are too hydrated, there's a number of things you can measure with a smartwatch, we think, that would have value. Won't be a clinical grade measurement, will be a tip-off, if you will, that something's off, much like the infectious disease. Now, we have an amazing cohort that actually does all kinds of things. They do an exercise study. They ran to their VO2 max. We can see all kinds of cool molecules change. And most recently, they've been doing a fiber study. I thought I'd put this in because if it's nice with some of the other speakers' work talked about today, you hear fiber is good for you. But, you know, if you think about which one, there's a zillion fibers out there. They're long chain, short chains, hydrophobic, hydrophilic, positive, negative charge, you know, which fiber is good for what. And if you look at the literature, it's all over the map. You know, some fibers are thought to be good for cholesterol, others for glucose, and so on and so forth. So we broke it down. We started taking purified fibers, just took the 2 most popular ones, arabinoxylan and inulin, and put people through, you know, increasing amounts for 1 week each. And they're randomized whether they're on arabinoxylan, inulin, and then we did a mixture of 5 fibers, including arabinoxylan. And then sure enough, you'll see thousands of molecules change. This is, you know, all the molecules that went up with arabinoxylan. These are the ones that go down, that go through a washout period. You can see them come back to normal. And it turns out that we found, this is stated in literature, and there's a fair amount of evidence for it, although you'll find contrary evidence as well, that, you know, in general with arabinoxylan, people's cholesterol will drop, LDL, the bad one, which is good. And mixture, it drops not as much, believe it or not. And inulin, now the inulin, you'll see it reported all kinds of different ways in the literature. You'll actually see some people say it drops cholesterol, other people say it doesn't. we didn't find a drop overall from our cohort. So inulin did not drop cholesterol, arabinoxylan did. Interestingly enough, we could actually see secondary bioacids change also go up. And we actually have a new model for how we think arabinoxylan works. We actually think it's metabolized and goes through these secondary bioacids, which is different from the model that's out there. Most people think cholesterol binds or gets bound to fiber and gets sucked out that way. We have a different model now based on this work. Just picking up on Abigail's comments, we can show different fibers actually stimulate. This is arabinoxylan. Oops, missing. This is inulin. Different fibers will stimulate the growth of different microbes. So that's not surprising, but it hasn't really been shown for purified fibers the way we're doing it. I forgot to emphasize, these are purified fibers. So these microbes grow with arabinoxylan, these with inulin. And perhaps this is the most important slide. Everyone is different. So even though I told you with arabinoxylan, the cholesterol goes down, that's true for this person, but here's a person, they didn't go down at all with arabinoxylan. And then yet with a different fiber, this inulin, they actually went down, their cholesterol went down with inulin. So different people were, in spite of what I told you as a population, individuals did behave differently. And this is the power, this is why we have to understand people at an individualized level. So to help do this, we built a personal health dashboard that pulls in all people's wearable data, microbiome, all kinds of omics data, and displays it on a smartphone. So we think a smartphone is going to be more important than a doctor in the future. Doctors won't go away. They're still very important. But they'll be aided with these smartphones. And then you can follow your health measurements at whatever resolution you want, daily, weekly, monthly, yearly, and follow what's going on. And so I see a very different world coming up where we can follow with big data people's health measurements at a personalized level and much, much better manage people's health. And so I have an amazing lab. I won't have time to go through this, but I have great collaborators with George Weinstock, Tracy McLaughlin. The Wearables team is shown here, and we now have a very large Wearables team. Do sign up for a study. We love data, as you can tell. And so now I think I'm done and we're ready for the panel.